All right, so like we said, we're going to be doing muscle tissue today. Okay. Oh, see if I can get this to. I don't know. I seem to always have technical difficulties all on the same day. It's just the way it goes with me. Okay. So muscle tissue. So this chapter, kind of like how we usually do, it's going to be, we're going to start with an outline and then we're going to go through the outline. So the main things that happen in this chapter is we have like a lot of stuff about muscles, and then we also cover muscle contraction. So one of the main points that we're going to talk about is the sliding filament mechanism, and this is how your muscle contracts. So that'll be a main part of the chapter. And then the other part of it, we're just going to call like anatomy, kind of anatomy of muscle. And then that gets broken down into quite a few topics from there. So we have our unique characteristics of muscle. So I'm going to see how much we can draw on this outline of stuff that's a little more simple. Okay, so for 4 characteristics, muscle is excitable. Okay, so what excitable means is it receives electrical stimulus from the nervous system. So under here, let's write stimulus from nervous system. 2, it's contractility. So it contracts. This means it shortens when stimulated, right? So if we have our muscle is contracting, it's shortening. Excitable, it's responding to a stimulus. Elasticity is another characteristic of muscle. This doesn't mean it stretches. This means it returns to its original shape. So when it's stretched, it comes back. Like, see if I have, like a ponytail, right? If you take your ponytail and you stretch it and then you return it, it'll come back to a circle. That's elasticity, okay? Versus extensibility, and extensibility is stretch. Extensibility, stretch. So when you actually stretch it, this would be, where is it? This is extensibility, elasticity when it comes back into a circle. That makes sense. So those are the 4 characteristics of muscle. You guys already know that skeletal muscle is striated and voluntary. I don't think that's necessary to cover again. You guys know that. Functions of skeletal muscle. Skeletal muscle functions to help move the body, to maintain posture. And this is in the fact that 10% of your muscles are contracted all the time. So 10% of muscles are always contracted. That's what maintains your posture. It helps regulate temperature. This is in the form of shivering, right? When you're cold, you shiver and that helps warm you up. And then it helps to store and move different minerals. So this is things like glucose and calcium. And then it also functions for support, right? Your muscle gives like structure to your whole body. So that's kind of our basics of functions of the muscular system, and then characteristics of muscle, like intro topics. Then we get into, so again, sliding filament mechanism, we'll kind of go through on a different page, but more anatomy. Muscle is divided into its basic components that kind of build up. So our smallest component, so we're going to go from, let me see, 1, 2, 3. So let's do extra small. I'm going to just put this is not like something that's on the test, but just to give you like a little perspective. So we would start with our myofilaments. This is your actin and your myosin. Myofilaments join together to become the next largest component, which is the myofibril. And on the next page, I'll make a drawing. Well, I guess I could try to do it on here. Let's see how that looks. The next one would be your myofiber. And then the next one is your fascicle. And then lastly, those join together to become your muscle. So I was just realizing, oh no. Oh. Wait, this is about to disappear. I don't know how that happened. Okay. Well, that was interesting. I think I must have. Let's hope that I accidentally hit something and that's not gonna happen to me again. So I don't know. I think it just kicked me off and then now I'm back. So hopefully it'll be fine. So I'll just draw them bigger with a picture because I feel like that's probably more clear anyway. So myofilaments are our actin and myosin. Those are going to be small. So if you're looking at a myofibril, along the surface of it is actin and myosin. So this would be an actin, myosin, actin, myosin, actin, myosin. So this entire thing here is our next size up, which is a myofibril. So each of these guys is a myofilament. They are the smallest component. And a key thing to know for the test is that they do not run the entire length of the muscle. So your myofilament, again, is the only structure that does not run the entire length of the muscle. Your myofibril does run the entire length of the muscle. And then our next size up, we take our multiple myofibrils and we get them together. And together they form our next size up, which is our actual muscle cell. And this is the myofiber. Okay, so multiple myofibrils become the myofiber, and this equals muscle cell, right? So myofiber is the muscle cell. Multiple muscle cells join together and they become the next size up, which is our fascicle. Okay. And the fascicle is, again, groups of myofibers. And then you actually get multiple fascicles that would then duplicate and become the final thing, which is our muscle. Groups of fascicles. So if you drew an entire muscle kind of crossways, inside of that would be fascicles like this. Okay. Each of these is a fascicle. Inside of the fascicle, myofibers. And then inside of the myofibers, each of those would have myofibrils, right? And then each of the myofibrils, like we said, breaks down and it looks like this. And along the surface of it, we could kind of draw different colors this time, is our actin and our myosin, right? And those are the myofilaments. Let me see if I have, okay, so now we talk about the coverings. Okay, so muscle is covered around by a connective tissue that's called dense, that's made of dense irregular, and this is called epimysium. So for the test, you want to know that this is dense, irregular, connective tissue, and that it is the covering around the entire muscle. So covering around the entire muscle. Does that make sense? So the muscle layer has epimysium. The fascicle has what's called perimysium. It is also dense irregular, and it would be the covering around each fascicle. Is that pretty clear? Around each fascicle. So we would put that here, perimycium. The myofiber also has a covering. This is called endomycium. It's going to be the same thing. It's made of connective tissue, but this is our big key difference. It's made of loose areolar. So the other two are made of dense irregular. Endomycium is made of loose areolar. Okay, so for the test, you definitely want to know dense irregular for the outer two and then loose areolar for endo. So that would be here, endomycium covering each myofiber or muscle cell. The myofibril has no connective tissue covering it. And then the myofilament, no connective tissue covering. So again, no connective tissue, CT. And on the myofilament, actin and myosin, no connective tissue. Okay. Does that make sense? Probably the picture on the right is easier to follow. You can kind of see it all. And you could draw one in your notes. If I was doing it on an outline, I'd probably draw a little mini version of that for like my memory's sake. I was going to try to draw that, but we were kind of out of room on the outline, so it worked out better. That didn't work. All right, and then there's two more layers. So I'm going to kind of erase this. So there's two more layers you need to know, and then we'll kind of get going some more into other things. So for the muscles themselves, right, we have our muscle and our muscle. Let's say you have your biceps and your triceps muscle. We're all pretty familiar with biceps and triceps if you go to the gym at all, right? So inside of each of our muscles, we have our fascicles, right? Like we drew on the right. Fascicles, inside of each fascicle, we have our myofibers. And then inside of each myofiber, we have our myofibrils with the myofilaments, right? Red dots. Separating each muscle from each other is a structure called, let's do it in yellow. It's called deep fascia, okay? So deep fascia, and this separates muscles from each other. Then we have, like, let's pretend that this is, let's just erase this here. Let's pretend that this is your biceps and your triceps, and on the surface here is your skin, right, with your hairs on it, on your arm. Separating the muscle from the skin is what's called the superficial fascia. Superficial fascia. Let's make sure we don't get confused with zero here. Okay. Superficial fascia separates muscle from skin. Both of the fascia would be like thick mycium. They're like a thick protective coating. I don't really think that I need to write that because it's not really what you need to know for the test. Okay, so this is probably, I would say this would be a good picture to have like in your notes or take a picture of if you're visual, because this probably kind of lays out all the layers kind of in one spot. Okay, any questions there? Does that make sense? Myofilaments to myofibril to myofiber, fascicle, muscle, and then all the different layers and coverings around each of them. All right, so kind of going back to some more general stuff, and then we'll get into contraction itself. So again, we were on like that muscle tissue outline, and we said there was anatomy. We did our 4 characteristics. We did some functions of muscle. And then what we just talked about maybe would be like, I don't really know exactly the word. Let's see if there's a word. All I have is anatomy, but I like layout, like smallest to largest, right? Getting really small to big. Other topics are the attachments of muscle. These are pretty simple. These are the same ones that if you've started on your muscle packet at all over the weekend, you're familiar with at this point. muscle has an origin, an insertion, action, and innervation, right? It's a pretty simple concept. For the test, remember you need to know definitions of these, okay? So for origin, it's the part of muscle that attaches to bone that does not move, or moves slightly. So that's origin. And you would want to memorize that definition just like that is what I would recommend. Insertion is the part attached to bone that does move. All right, so like we said, origin does not move or move slightly, insertion does move. Action is the movement. So like if you flex your arm, extend your arm, this would be like example flexion, extension. Innervation is the nerve supply. So the brachial nerve supplies the bicep. For the origin of the bicep would be the humerus, right? Like the top of the humerus. The insertion of the bicep muscle would be the radius. Does that kind of make sense? Origin, where it attaches that doesn't move, top of the humerus, and then insertion where it does move, so it's what's moving your arm and pulling it upward. So for the test, you may get a question, something like this, you know, which of these would be the insertion like description of your biceps? Is it the part that does move? Is it the part that does not move? But you're not going to be getting into like examples, like maybe the example your professor gave you when he instructed this, but other than that, you won't get anything else on the test. So you're not going to be doing pectoralis major and all these other muscles that you are required to do for the packet, right? Reminder, you have to write the origin, insertion, action, and innervation in your own handwriting for receiving full points on the muscle packet. But you are not going to be tested on origin, insertion, action, innervation for the test, whether that would be lab exam or lecture exam. What you are tested on is the definitions. So what we're going to write is definitions only for test. Good, and it would be these exact definitions that I just wrote down. Okay, so a few more kind of things in terms of the anatomy of a muscle cell. You have your muscle cell, so we'll kind of remember that there was the muscle, and then inside we had our little fascicles, right? And then within each of those fascicles, there was our little myofibers. So the myofiber is the muscle cell. And inside the myofiber or the muscle cell, like we had said, there are those different myofibrils, right? And this is where we're going to be spending our time when we talk about contraction. So there was the myofibril, and then along the surface of one myofibril, we said that it had what? Actin and myosin, right? So we'll do myosin in black, actin in red, actin and myosin. All of the area in between, okay, all of this area here is our sarcoplasm. Remember the cytoplasm of a cell, the cytoplasm of muscle cell is sarcoplasm. And then it also has its own endoplasmic reticulum, which stores calcium. We'll kind of just draw that like in here. I'll draw another picture and show you guys on another screen. But sarcoplasmic reticulum is going to be the endoplasmic reticulum of the muscle cell, and it stores calcium. So that's really important to know for the test. And then lastly, you have your plasma membrane of a muscle cell. All right, so the plasma membrane. And this is called the sarcolemma. Remember, your myofiber is covered by endomysium, and endomysium is loose areolar. So in a sense, the myofiber has like two coverings. It has the sarcolemma, and then it also has the endomysium, and I'll kind of draw that squiggly. Is that making sense? So kind of two different things. Endomysium and sarcolemma both cover that muscle cell. And like we said, sarcoplasm and sarcoplasmic reticulum. Any questions here? I saw. Right. Yeah. Someone asked, you only do origin, insertion, action, and innervation for the muscles that you label. Right. You're not going to do it for veins or a bone, right? Because this origin, insertion, action, and innervation, that would only apply to your muscles, right? It's not going to apply to your bone. Good question. All right, so we'll talk about muscle contraction in just a second. I kind of want to do that until we end. So what I'm going to do is I'm going to just jump to the part of the chapter that's some more terms, if that's okay with everybody, and then we'll come back to contraction, if that works. So the rest of the terms for the chapter will kind of just go over those because I feel like they're all kind of makes sense to do them on this general page. So a motor unit, is just a very simple definition. It refers to a single motor neuron and the muscle fibers. Again, muscle fiber is the same as myofiber, which is the same as muscle cell. So there's three names for a muscle cell. Muscle cell, myofiber, or muscle fibers. One of the things that you guys seem to kind of get thrown off on the exam is like if you see a different word or a term and you didn't memorize that, you feel kind of thrown off. So just make sure you guys study those, either write them down a few times, but remember a muscle fiber is the same as a myofiber, and a myofiber is the same as a muscle cell. So we could use those interchangeably on a question, either way of those. So when we refer to a single motor unit, that would be the motor neuron and the muscle fibers it connects. So we're going to talk about that, but remember a neuron kind of looks like this. So if this is a motor neuron, we're going to be looking at this motor neuron. And then let's say that this is my myofiber here, right? And this little motor neuron sends its signals to each of these myofibers. Does that make sense? So that would be one motor unit. It's pretty simple. It's just the definition that you would need to know. I'm going to erase this, kind of drop it. Then we have muscle tone. Okay, so for muscle tone, this is broken down into two types of muscle contraction. So we have our muscle tone, which is our constant tension in the muscle. Then we have muscle contraction. And there are two types of muscle contraction, and they're pretty simple, but you do need to know them and then their example. So you have your number one, which is our isometric. Isometric contraction is when the force is less than the load, which results in no movement. Okay, isotonic, this is when our force is greater than the load and we get movement. So when you go to the gym and you lift weights, you are performing isotonic contraction, right? You're able to lift it. If you go to the gym and you try to pick up 400-pound dumbbells, right? You're not going to be able to lift those. That's isometric. The way that students have said they remember isometric is it's a straight line like a meter stick, or a meter is long, so you can't contract, can't lift up, right? Admit. So I'm trying to admit this person, assign the person to the breakout room, assign to here. Okay. Whereas isotonic, you can overcome it. There are two types of isotonic. So there's concentric, and eccentric. Eccentric is when you're lengthening that muscle. Lengthen. Maybe you could think of it like extend. Extending out the muscle is eccentric. Concentric is when you shorten that muscle, so you bring it back. Concentric, like consolidate or bring it back to you. So lengthening that muscle, lengthen muscle. Eccentric, shorten muscle, concentric. For your types of skeletal muscle fibers, let's see if I can just do this really quick. So types of fibers. So just what you need to know for the test here, you have your slow oxidative fibers. Main thing to know is this would be your red muscle, and then your fast glycolytic fibers would be your white muscle. Red muscle would be for long movement, right? Like a duck. This is when he talks about the chicken flapping its wing versus the duck. And then white muscle would be for quick movement. And that's all you need to know for that. Okay, slow oxidative is red for long movements, like sustained movements. Fast glycolytic for quick movements. You won't really see there's 4 different patterns of fascicle arrangement: circular, parallel, convergent, and pennate. You're not really going to see that on the test. So I'm not going to write them down, okay? Because they're kind of like, did everybody hear that? There's like 4 patterns of fascicle arrangement: circular, parallel, convergent, and pennate. There's unipennate, bipennate, and multipennate. It might be one question. Please, I would just recommend don't spend time on that. That's like a waste of your time. I don't remember a question on that, so the likelihood is on there is very slim. And it's kind of complicated, so I just wouldn't study it. All right, so let's talk about exercise and skeletal muscle, because there will be questions on that, and we'll do our actions really quick. And then, like I said, after that, we're just going to focus on muscle contraction, which is where there's a lot of questions on the test and it gets more confusing. So we want to go to that next, but we're just kind of making sure you guys get your basic outline of the chapter first. Okay, so we did our 4 characteristics: functions, layout, attachments, motor units, muscle tone, muscle contraction, types of fibers. And then we talked about our anatomy of it, and then we'll talk about exercise and some actions, and then we'll go back, okay? Mnemonic for concentric, eccentric. I would say the way I would remember eccentric is like extension. You're extending out that muscle. Concentric is you're pulling it back. The way I do remember this might not help anybody, but it's how I remembered it. An isotonic is like you're drinking a gin and tonic. And if you picture how lightweight a glass with gin and tonic is, everybody's able to pick up a glass of gin and tonic. Does that make sense? So isotonic, it's really, really easy to pick up a gin and tonic. Isometric, you can't pick that weight up, it's too heavy. And then with isotonic, with the gin and tonic, I thought of like eccentric, you're reaching out to get that glass. Concentric, you're bringing it back to yourself. Does that make sense? That's how I remembered the 2 when I was in the class. Eccentric, extending out for my glass of gin and tonic, and then concentric, pulling it back. Okay, so for exercise and skeletal muscle, so you have trophy. Trophy is cell size. So if we have hyper, there's an increase in cell size. I'm just going to keep this simple because I think you went over this in Unit 1. If you have hypotrophy, it's a decrease. So again, hypotrophy would be a decrease in cell size. Hypertrophy would be an increase in cell size. The word hyper means increase, hypo means decrease. For plasia plasia means cell number. So if I have hyperplasia, I have an increase in cell number. If I have hypoplasia, I have a decrease in cell number. Does that make sense? If you just memorize plasia, number, trophy, size. How I remember this is when somebody has like a trophy, you know, comes home and they're carrying a trophy and they're so proud of their trophy they won. They are like maybe buff and have really big arms, like big cell size, versus plasia is number. So trophy, if I can remember that size, I know the other one's number. That's kind of how I remember those two. Okay, atrophy. So atrophy is when we have both together. So atrophy is hypotrophy plus hypoplasia. So this is when a person has a decrease in cell size and number. This usually is a patient that's like, let's pretend that this patient has been laying in bed, right, in the hospital for weeks, and they haven't been moving their muscles. Now they're losing their cell size and number because they're just laying in bed. Their muscles are atrophying, okay? And this is because of a lack of stimulation. This is what happens to people like when they get paralyzed and they're in a wheelchair, they don't contract their muscles again and they atrophy. Versus hypertonia. Hypertonia is an increase in muscle tone. So this is someone who has rigid muscles. And then we have hypotonia. Hypotonia is a decrease in muscle tone. And the example here is floppy baby syndrome. It's like babies who don't have any muscle tone in their neck or their muscles. We have metaplasia. This is a change from one tissue type to another. And this occurs when we change from ciliated, pseudostratified, columnar epithelium. This is an example you definitely want to know. So you change from ciliated, pseudostratified columnar epithelium to keratinized stratified squamous. And this happens to smokers. Lastly, we have neoplasia. Neoplasia is out of control growth, which would be cancer. And then our very last thing, and then we're going to get into our contraction. So we have our actions. So actions of muscle. This would be our agonist and our antagonist and our synergist, all right? So hang with me, we're almost there, and then we're going to go over to contraction. So agonist is the one that we call the prime mover. This is the one that performs the action. So for example, your biceps would be doing the flexion. Antagonist opposes the agonist. So for example, the triceps is doing extension, right? So they're opposing that biceps. And then the synergist helps the agonist. So our example is the brachialis muscle would help the biceps. Just know for the test that they can reverse. If you're talking about extension, the triceps would become the agonist because it's the one doing the action, and the antagonist would be the biceps because it opposes it. There's a few things about cardiac and smooth muscle, but those are very simple, and I believe you guys have already gone over those in Unit 1. So I'm not going to spend time on those. I want to start our discussion on contraction. Okay, so we're going to go into that. Any questions on this? This is just kind of terms. These will all be, there are multiple of these on the test for sure. So I would definitely spend some time going over these if you haven't already, okay? Okay, so when we talk about our contraction, the first thing that we cover in this class is what contraction looks like under a microscope. So you're going to learn some different terms and areas that you need to focus on, and then I'll try to make that very simple. And then we will also talk about what happens at the cellular level. So first we'll look at the microscope, and then we're going to go to the actual sarcomere inside the cell. So those are the two main areas we'll talk about. And then after we talk about the sarcomere, we'll talk about how the nervous system communicates with muscle. Okay? So that's why it gets a little bit more in depth, but we start with the microscope. So when you're looking at the contractile units under the microscope, we have these are called Z-discs. Z-discs mark the outer limit of the sarcomere. So they're kind of just like a boundary, the Z-discs. Then we have our center, which is our myosin. This is a representation of what you see on the microscope. It's not like an exact drawing, it's just supposed to be a representation. And remember that the myosin is your myofilament. It's the dark A band. Then we said that we have our actin. Okay, actin. And that's also, right, a myofilament, and the actin is your light I band. Holding the myosin in place is titin. Titin is another one of our protein filaments here. Titin. Definition for your test: holds the myosin in place. Then we have our different zones on the microscope. Okay, so I just showed you the terms. Now it's your zones or your areas on the microscope, and then I'll make sure I write the definitions down for that. So you have what's called your light or your I band. And this is the definition you knew before, is that the actin or myofilament is your light or I band. Technically, that is not 100%. So we'll show you exactly where that would be. So this is your light or your I band. Like I showed you, it's this area here in the center. And the definition of the light or I band is actually where there is no myosin. That is your correct definition. So if you look, I'll try to draw it like this. Okay. So let's look here real quick. I'm going to draw a little arrow. Do you guys see how if I draw a line here and here, everything in between is the dark or A band? So I will do a new color. Okay, here we go. So everything in between the red lines here, this is the dark or A band. Do you guys see how there is a little bit of actin still left inside of that dark A band? I kind of drew some arrows pointing at it. So actin is just the myofilament that is basically a different myofilament than myosin. The correct definition of light or I band is where there is no myosin. So on the first unit, when we told you guys that the light I band is actin, that's not really correct. I kind of wish we didn't tell you guys that. The correct terminology is actually where there is no myosin. Now the dark A band is the length of myosin. So when we told you that myosin is the dark A band, that is correct. But the light I band is not actin. It's the correct definition is where there is no myosin. Then we also have the H zone. The H zone is the area where there is no actin or myosin, okay? So kind of how I showed you that there's still some actin in here. If we kind of erase my little arrows here, and we'll do green. This part down the center where there's no actin and there's only myosin, right here, that's my H zone. H zone is where there is no actin, only myosin. So you see how in the dark A band, it's the length of myosin. Some of that A band has actin, some of it doesn't. But that H zone, or the area in the middle, is where there is no actin and there's only myosin. So the way I think of it is it's kind of like a shadow in a room. The dark A band is like it casts a shadow, but the actin makes that shadow even darker. So if you kind of look at it, there's two different colors. There's an area where it's really dark and there's an area where it's lighter. And this is, again, reminding you, this is all the stuff on the microscope. So that's why sometimes it's a little hard to picture. But that's why we look at these different terms, and then I'll make sure it's clear what you need to know for the test. And then finally, the M line is just the middle line. So this is what scientists would look at on the microscope to tell if something's contracting. All right, so now let's talk about what changes or shortens in the microscope when they watch a muscle contract. So what they do is they have a little live, I'm not entirely sure how they do this, but somehow they have a live view of the muscle and they apply a stimulus to it and watch it contract. And when it contracts, certain things change. So the Z discs do not change or shorten when the muscle contracts. So on your exam, you're going to be asked which of these changes or shortens when the muscle contracts. And you're probably going to get, I would say, 2 to 3 to 4 questions on this. I'm not entirely sure the number, but you will get a few questions on this. So the Z discs do not change or shorten when the muscle contracts. That makes sense because they're just like the border or the outside, so they're not going to change or shorten. The dark or the A band does not change or shorten, does not change or shorten when the muscle contracts. It just stays in place, basically. The titin is not going to, the titin actually will probably be, since it's part of the I band, never mind. The M line will not change when the muscle contracts, okay? The light or the I band is what is going to actually move towards the side. So when the muscle contracts, you have myosin at the center and actin kind of comes towards the middle. So what happens if I draw like a big arrow, this actin moves itself towards the center. So what we see is a shift where everything gets very short and narrow. So where before those actins were like way out here, as muscle contraction happens, they actually push and overlap at the center, if that's kind of making sense. So everything pushes and overlaps towards the middle. So the big thing you would want to make sure you know is that that light or I band does change or shorten during contraction, okay, because it's going towards the middle, and that H zone does change or shorten. In fact, the H zone disappears when the muscle contracts. And that's because as that stuff, like I said, starts to shrink towards the middle, we erase this, that H zone will actually disappear because there will be actin everywhere. Again, definition of the H zone is the presence of no actin at all. So if the actin is moving towards the middle and overlapping, the H zone disappears. Does that make sense? Our light or our I bands shorten up, our H zone disappears, our dark A band stays right there in the center as everything moves towards the middle, okay? And then we have that titin, which is basically not something you need to worry about. And then I'll give you one little kind of quiz thing and see if you guys kind of get the idea. So this would be one sarcomere, okay? And a sarcomere is the contractile unit of the muscle cell. So if this is the sarcomere or contractile unit of the muscle cell, does this change? Well, maybe I should do it as a question. this changes with muscle contraction, like I've been saying for each one. Does it change or shorten? So is it true or is it false? Does the sarcomere change or shorten with muscle contraction? So put a true or a false in the chat for that. Good. Okay. So you guys are saying true, right? So just a quick recap, just because I know this is kind of a little bit tricky. So the best analogy that I've found for explaining this, it's not like perfect, but if you picture that my hands are the Z discs and both hands together is a sarcomere, right? So the thumbs out here are my Z disc or my peripheral. My myosin is kind of like my fingers here. And actin is my palms of my hands, right? So as we have, it's called the sliding filament theory, because the filaments will slide together as we contract. So if my hands are the sarcomere and I cause contraction to occur, actin, which is like the palms of my hands, move towards the center, right? And so that's going to cause the shortening or contracting of the sarcomere. Does that make sense? Z discs would not change, right? The dark or A band would kind of be like that darkness at the center that's not changing, okay? And the actin is going towards the middle, so that those light I bands are changing. The H zone, if I kind of left a little gap in between my fingers here, we could say that's the H zone. That would disappear as my fingers join together. Hopefully that kind of helped a little bit. You can kind of picture that coming together and getting shortened, okay? All right, so that was under the microscope. So there's some different things there that you would need to know. Like I said, you want to know your term, your definition, and then whether or not it changes or shortens when the muscle contracts. So those are kind of the three things you need to know: term, definition, and whether or not it changes or shortens when the muscle contracts. Good. All right, so let's talk about our next part. So the sarcomere inside of the cell. So the contractile unit inside of the muscle cell. So there's this kind of this way that we draw it. So this is going to be our first protein filament. It's called tropomyosin. Attached to the tropomyosin are little additional protein filaments we call troponin. Okay, so troponin. Probably going to need to, let me see if I can. Okay. All right, and these are troponin here, troponin. And then we have little balls of actin. Oh, I'm doing the wrong color. See, what color did I do for actin? Yellow. Okay. Balls of actin. And this is going to be contraction. I'll write a little step by step because this would be where the exam gets kind of challenging. So actin, as you already know. And then we have our myosin, right? And myosin is waiting out here to perform contraction. Okay, like we said, it wants to do that sliding filament mechanism like I just showed you with my hands. It wants to contract the muscle. So myosin is waiting out here. Okay, so troponin is attached to tropomyosin, and tropomyosin is right here. The main thing that tropomyosin does is it covers the active sites on actin. So each of these little actin heads has a little active site, and that active site wants to attach to myosin. So active site on actin wants to attach to myosin. I always feel like this whole contraction thing really does feel like, you know, Romeo and Juliet. It's like actin and myosin are in love, and actin has these active sites, but tropomyosin is covering those active sites up and keeping actin and myosin from being together. Okay, so what this whole process is how do we get to the story of contraction is how do you get tropomyosin off of the active sites on actin so that myosin and actin can finally be together. That's kind of the whole story of this. So down the center, we have what's called a T tubule. I didn't really leave myself room for it, but just imagine that these little ones are gone. So we have these transverse or T tubule, transverse tubule stands for T tubule, okay? And this allows sodium to come in to the muscle cell. That sodium is coming from the nervous system, and we're going to talk about that as well. Now, hanging off of the side of the T tubule is sarcoplasmic reticulum. Sarcoplasmic reticulum stores calcium. And finally, and then we'll do our quick step-by-step, we have on the edge of that sarcoplasmic reticulum, we have little gates called terminal cisterna. Okay, these are gates that allow calcium to leave. All right, so our step number, let's see if I can kind of draw this. This will be our goal, is to bend that tropomyosin away and expose our little active sites on actin so that myosin and actin can finally be together. So that's going to be our goal. And then we'll do a little step-by-step of how we get there. All right, so step number 1, sodium comes down the T tubule. That's step number 1. When sodium comes down, it triggers calcium to leave the sarcoplasmic reticulum. So the entrance of sodium here, you can see me drawing over here. When the sodium enters that T tubule, calcium molecules, let's see, let's do them in blue, let's do them in red. Little calcium molecules start to leave that sarcoplasmic reticulum. They leave out the terminal cisterna, which are little gates. So we're going to leave the sarcoplasmic reticulum, out the terminal cisterna. So that's where our calcium leave. Step 3, calcium then goes and binds to troponin. So that little calcium molecule, each of those little calcium molecules, they're going to go out here. So I'm writing Ca, well, we can just spell it, calcium, calcium, Ca, they're attaching to those little troponins, calcium attaching to all those little troponins, okay? Step 4, when they bind, they change troponin's shape. Changing troponin's shape then changes the shape of tropomyosin. So it's kind of like tropomyosin and troponin are bound together, right? And when we change troponin's shape, we change tropomyosin's shape, 5. This then will bend tropomyosin away from the active sites on actin, away, and this exposes the active sites on actin. Like I said, those actin molecules are like Romeo and Juliet, right? They're kept apart from the myosin, but as soon as they're exposed and that tropomyosin is bent away, we have step 6, the active sites of actin form a cross bridge with myosin, with the myosin heads, which causes them being the myosin heads, to contract, and we have contraction of the muscle. So how do we get contraction of the muscle? When myosin and actin are finally allowed to be together. How do we get myosin and actin together? We start with the sodium coming down the T-tubule. That triggers the calcium to leave the sarcoplasmic reticulum out of the terminal cisternae, which are like little gates. When that calcium leaves, it binds to troponin. When that binds to troponin, it changes the shape of tropomyosin. When tropomyosin is taken away, actin and myosin can finally be together, and we call that a cross bridge, and a cross bridge results in muscle contraction. All right? So that is the first thing you need to know about muscle contraction, right? So it's kind of like the step-by-step process. That's definitely the like nice, fast, simplified format. I really recommend you listening to your lecture because it's probably going to have even more details, but hopefully that's a really good review. But what we're going to continue talking about is how the nervous system communicates, okay? So how the nervous system communicates and gets that sodium. So like we said, sodium comes down the T-tubule. How? From a motor neuron. So what you also need to know for your test is how does the motor neuron send the signal to the T-tubule? How does it get to the T-tubule? And then one more function or a structure here that you need to know is a triad. A triad is one T-tubule and two terminal cisterna. One T-tubule and two terminal cisterna. You can probably remember it as like a lot of T's, triad, T. That causes or allows continuous muscle contraction from top to bottom. So because of that triad, we make sure that we keep contracting all the way down from the top to the bottom. Okay? You already got all of those things. All right, and we will talk about how that nervous system communicates with the muscle. Any questions on this page? Make sense? All right, so we'll do how it communicates, and then we'll also do a little step-by-step. So we have your, I'm going to try to draw like a little zoomed out version, and then we'll go closer. So here's your neuron, right? Has that cell. This is like a recap in case you forgot. Dendrite, cell body, axon, hillock, axon. Do you remember myelin sheath? Yes? You need to know this again for this exam. These were what? Telodendria. And at the end of every telodendria, you had an axon knob, synaptic knob. Sometimes we also called it an axon terminal, right? So where this whole entire thing is just the anatomy of a neuron. It could be a motor neuron or it could be a neuron. We're just saying neuron. Now, when it communicates, this neuron, it can either communicate with the dendrites of another neuron, okay? And I know they look a little different than, my drawing looks a little different than the first guy, but that's just to show you how they connect. So when one neuron communicates with another neuron, they communicate with the dendrites, right? Dendrite to telodendria. On your test, we're going to talk about how neurons communicate, neuron to neuron, and how neuron communicates to muscle. So you kind of need to know two things. Neuron to neuron, we call that a neurosynapse. And then you also need to know neuron to muscle. Neuron to muscle is called a neuromuscular junction. It's going to be the same concept both times, where they communicate with each other. Neurosynapse, neuromuscular junction. Okay. So let's talk about, trying to figure out which one makes sense to talk about first. Really doesn't matter, honestly. Let's talk about the neuromuscular junction first, because that's what we were just on. So in a neuromuscular junction, what we do when we talk about this concept is we just draw for you guys the telodendria and then our synaptic knob. I just kind of wanted to make sure everybody get where we are right here, and then we draw it expanded. Does that make sense? But I'm going to need to erase everything on the left. I'm just reminding you of the anatomy. So when that telodendria and synaptic knob of this time, this would be of a motor neuron. And a motor neuron is just a neuron that communicates, neuron that sends signals to muscle. So it sounds like it's something special. It's not really, it's just one that communicates to muscle. And then we have our motor end plate, okay? I had kind of shown you on the model, this is the wavy portion of the sarcolemma, wavy portion of the sarcolemma. And remember, the sarcolemma is the plasma membrane of the muscle cell. And our muscle cell is also called myofiber, right? So I'm going to write sarcolemma, plasma membrane of myofiber, and you guys remember that means muscle cell. So the wavy portion of the muscle cell, and it is wavy to assist the T tubules in reaching all the way through the muscle. So each of these coming off of it would be a T tubule. Okay, and remember that coming off of that T tubule is the sarcoplasmic reticulum. And what does the sarcoplasmic reticulum store? Little calcium molecules, right? So calcium, sarcoplasmic reticulum. What was on the edges of each sarcoplasmic reticulum? Terminal cisternae, right? And then what about the T tubule? T tubule. And then we remember that that whole thing was like connected, or was basically a part of our contractile unit, right? And we had those kind of like this, right? Trying to like draw it all in one place so you guys can see it. And those were our little actin balls. Yes, the stuff we just went over. Okay, so our motor end plate is wavy. It's wavy to assist the T tubules in getting down into that muscle. All right, hopefully everybody is with me at this point. So what's going to happen first that we talked about before? Sodium gets into the T tubule. But the question that we have is, how does sodium get into the T tubule? And that's what we're going to talk about, okay? So our first step actually starts with sodium again. So I'm gonna do this one in green. Everybody can read that? Yeah, okay. So step 1, sodium travels down the axon and then the telodendria of the motor neuron. All right, so when you think something, like you think I want to move my leg right now, right? I'm moving my leg. When I think I want to move my leg, the way that the brain does that is it sends sodium, which is abbreviated Na, right? It sends it down its neurons. Here's a neuron. And eventually it gets out to a motor neuron. And that motor neuron will actually communicate with the muscle. Does that make sense? So sodium, this is sodium. Sodium travels down that telodendria. I think I missed some different letters in that. Telodendria. Okay, sodium travels down. That's step number 1. Step number 2. When sodium enters the synaptic knob, it causes calcium to come inside the synaptic knob from the extracellular environment. So this probably, hopefully you guys are following me, but this is probably feeling a little bit overwhelming. This is kind of an overwhelming area of the chapter. It's not all this hard on this unit, but just know that this is a harder unit. Some of the concepts get a little more tricky like this stuff. It's probably feeling a little more like physiology to you guys. Okay, so calcium comes in. This is calcium. So sodium is going to be step 1. Calcium is really step 2. And sitting inside of this synaptic knob, we have what are called synaptic vesicles. Synaptic vesicles. And a synaptic vesicles store neurotransmitters. They store neurotransmitters. There are a variety of types of neurotransmitters, but the neurotransmitter for muscle contraction, and the one we will be testing you guys on, is called acetylcholine. Acetylcholine is the neurotransmitter for muscle contraction. It's abbreviated ACH, acetylcholine. So let's make that little black dots. Acetylcholine neurotransmitters, okay? So when calcium gets in, so let's go back over here. So when calcium enters that synaptic knob, it causes the neurotransmitters to break free or be released from the synaptic vesicles because the synaptic vesicles dissolve. So it causes the neurotransmitters to be released from the synaptic vesicles because the synaptic vesicles like pretty much dissolve. Okay, so number 3, step 3 is like all of those little neurotransmitters like getting out. So we're going to draw a little number 3 here. They kind of get out. Number 1, sodium comes down. Number 2, calcium enters into the actual synaptic knob from the outside. That's step number 2. And then step number 3, these little neurotransmitters get out. And what they're going to do is they're going to actually come over and get out of that synaptic knob and cross the synaptic cleft. Okay, so the space right here, the space is called the synaptic cleft. It's the space between the dendrite and that telodendria is the synaptic cleft. All right, so step number 4, the neurotransmitters cross over the synaptic cleft. Okay, so that's step number 4. So we'll draw number 4, crossing the synaptic cleft right here. Step number 5, the neurotransmitter. So it's acetylcholine, right? Abbreviated ACH. The neurotransmitter acetylcholine attaches to a neuroreceptor that is blocking the T-tubule. So on the surface of each of these little T-tubules, I'll draw it in blue, there is, let's say, like a little ball-like structure that blocks the T-tubule. So nothing can get inside, kind of like a cork. But when that neurotransmitter comes over, it's going to attach to the neuroreceptor and it's going to unblock that cork. So this is ACH, and each of these is what's called a neuroreceptor. When the ACH and the neuroreceptor get together, it unblocks or opens the channel. So number 6, this opens the channel so that sodium from the synaptic cleft, so the synaptic cleft has sodium in it. It's just kind of, let me do red so that it sticks out. All of these little red dots, oh wait, I did green before. All of the little green dots represent sodium. And as soon as we get this little channel opened up here, if I can erase this little guy. Of course, it erases everything. It's always the way it is. Okay, so as soon as that gets opened up, my little sodium molecules come in to my T-tubule. So number 6, step number 6, kind of you can't really see it very well, but step number 5 was over here, the binding together. Step number 6, this opens the channel so that sodium from the synaptic cleft enters the T-tubule to start muscle contraction. Does that make sense? It's very long. I know it's very long, but hopefully you can see it all in one place. So first we get sodium coming down the telodendria. That causes calcium to get in from the outside. The calcium breaks up those little synaptic vesicles, making the neurotransmitters leak out. The neurotransmitters cross the synaptic cleft and attach to a neuroreceptor. That opens the gate that allows sodium to come into the T-tubule. Once the sodium is inside the T-tubule, we can go back to this page and get these 6 steps. So once sodium gets into the T-tubule, it triggers the calcium to go out and bind to troponin, bending away tropomyosin, attaching the actins to the myosin, and resulting in contraction. This is really when you're like, how on earth could human beings be an accident? It's like pretty crazy, right? Like 15 steps just to get muscle contraction, which we do all day. So I think it's really cool. Like I said, starts with sodium, and then we go through the steps. All right, so the next thing that we cover is neuron to neuron. So this was neuron to muscle. Neuron to neuron is 90% the same thing, so don't panic. It's going to be like almost the same stuff. It's actually like almost exactly the same, just a few little things that are slightly different. Okay, so you're over the worst of it. This page, like I said, and then you want to go over this as well. Okay. Any questions there before we jump into neuro? So neuro is going to be the same thing again, mostly. So here's neuro. So we have our telodendria, our synaptic knob. This time we have our dendrite. Just so you know, I didn't say this before in so many words, but this is called our presynaptic membrane. This is the synaptic knob, is our presynaptic membrane. And then we have our postsynaptic membrane. And the postsynaptic membrane can either be a dendrite of another neuron. That's option number 1. Option number 2 is it can be the motor end plate. And the motor end plate of the sarcolemma, which we just talked about, right? So either a dendrite of another neuron or the motor end plate. So presynaptic, postsynaptic. Again, this would be our telodendria. And we already know this is a synaptic knob. Inside, we have our little synaptic vesicles. Again, what do synaptic vesicles have inside? Neurotransmitter, right? Just so you know, there are technically 6 different types of neurotransmitter. That's not on the test. On the test, all you need to know is acetylcholine. I just remember when I got to physiology, I was kind of surprised when I found out there was more than 1 type. So there are actually 6. Acetylcholine is just the most common. All right, and so we're going to follow the same process again. We have little neuroreceptors. I'll try to kind of draw them the same way I did before. So these are my little neuroreceptor. They are specific to acetylcholine, meaning when acetylcholine comes into contact with them, they will allow the opening of that gate, right? Synaptic cleft, we already said, is that space in between. And we know that sitting in that space already is sodium. And we know that sitting out here is calcium. So we're going to go through our little steps. So step 1, sodium comes down the telodendria. Probably not going to write it in as much detail as before. Here's my sodium. Step 2, calcium enters the synaptic knob. Right? Step 2, calcium enters right here. Okay. Step 3, that triggers breakage of synaptic vesicles. And when those little synaptic vesicles break open, it releases that neurotransmitter, right? The neurotransmitter goes across the synaptic cleft. Step 5, it binds to a neuroreceptor, right? So it breaks open, and then they travel across and they bind to that neuroreceptor. So number 3 is kind of when they like break here. Number 4, they travel. And then number 5, we get the actual attachment. 6, once they are actually attached, this opens the channel. It's technically a channel. And this allows sodium to enter the postsynaptic membrane from the outside, from the extracellular. Okay, so when that sodium gets in, it causes continued depolarization. Sodium then continues down the cell body, the axon, the telodendria, the synaptic knob, to the next neuron. So what we see with neurons is a connection between billions upon billions of neurons. So neurons are really, really cool because we can have kind of an endless connection of them. So even though you don't have the ability to form new neurons, what we don't really talk about a lot in this class, but is really cool, is you can form an endless amount of connections between neurons. So the way that all of those neurons communicate is sodium. So it all starts with sodium. And then if you kind of remember this little process, when you guys get to physiology, it's going to become more complicated than this. But this is a really good intro into kind of getting your feet wet and your wheels turning. If you can remember this and you can really get a good foundation here, when you get to physio, it's not going to feel as complicated because you have a really good foundation of what's happening overall. All right, so a few last things. So first, what is it that stops this process, right? So I told you that, we have that signal. So one of the things we could do is we could stop the signal, right? So stop that signal, stop telling your brain to contract your leg, and it'll just stop, right, as far as we know. But the one thing that you could think about is, okay, well, we know that that would stop sodium from coming down. But what about all of those neurotransmitters that are sitting in that synaptic cleft, right? Just kind of hanging out. What happens to them? Well, if we leave those neurotransmitters just sitting there, they will continue that signal until all of them are gone. Okay? So what we have is an enzyme called acetylcholine esterase. Can you guys read that? It ends in esterase. It is an enzyme that digests all the acetylcholine in the synaptic cleft after the signal is over. So after we're done contracting that muscle, in order to stop the process, we want to bring that acetylcholine esterase in so we can stop that contraction. Let's see. There is a snake. I believe this is how that. I'm blanking a little bit, but I think this is the action. So there's a snake that has venom. Poisonous venom, and the way that the venom works to kill the victim is it prevents like the breakdown of the neurotransmitter. So what happens is this person remains contracted until they die. It's like does that make sense? It's like you seize, you over contract. So without an ability to stop the contraction, this could become really negative. I think that they think this is what Parkinson's tremors are caused by, because there's like over contraction and potential like problems with like destroying the neurotransmitters in the space. I don't know if that makes sense. So what happens is you have this continual muscle contraction because there's no stopping it. In the brain, this could be a good thing. Like if you have serotonin that makes you feel happy, right? And we create a medication, which is most antidepressant medications act this way. And the medication stops the reuptake or the breakdown of the serotonin. You get more serotonin effects, which makes you happier. But for muscles, this could be really bad, right? Because prolonged contraction could cause exhaustion and death. Does that kind of make sense? So acetylcholinesterase is important. It will be on your test. Nothing I just said will be on your test. It's just always showing you how cool it is. I think it's cool. Acetylcholinesterase is the enzyme that stops all of this because it digests the acetylcholine. so that you don't have just continued contraction after contraction after contraction. There are 3 pieces of energy, and I didn't include this on the previous one because I wanted to get into that. But if you're looking at this whole entire process on the page I just pulled up, the sodium, the calcium, at no point do we need ATP or cellular energy. So this does not, so no ATP is needed for contraction. But we do need 3 ATPs to undo a contraction. I'm just going to try to write them right here. So number 1, the first ATP breaks the cross bridge. And remember, the cross bridge is this right here. So that's the first ATP. ATP number 1 goes in and breaks that cross bridge. ATP number 2 recocks, which recocking means to like prepare, re-prepares, recocks, like cocking a gun, gets it ready to fire. Does that make sense? Recocks the myosin heads, so gets those myosin heads ready to fire again, would be number 2, the second ATP. And then the third ATP pumps the calcium back into the sarcoplasmic reticulum. So on your test, you would want to know that no ATP is needed to perform a contraction for the muscle. 3 ATPs are needed to undo it. And then like I just wrote for you, what are those 3 ATPs and what do they actually do, each of them? And I wrote those down. Okay. All right, so again, muscle contraction, the steps leading up to muscle contraction in the brain itself and the nervous system, and then the steps leading up to muscle contraction at the neuromuscular synapse, and then finally, the actual contraction of the muscle. Okay, and that should be your whole muscle unit. And I actually finished with a little bit of time. So we'll stop there. And I'll let you guys go. If you have questions, please stay. If not, I will see you Thursday, 4:45 to 6:45. Okay, and we're going to go over nervous tissue Thursday and probably some models. Thank you guys.